c-MYC, the future of cancer therapeutics. As the population in Europe and the United States grows older, cancer will have to be faced by an increasing number of people at some stage in their lives. Modern biomedical research has achieved much to identify the underlying genetic problems. Nowadays, many scientists believe that each type of cancer constitutes a separate disease, and therefore has to be treated with separate therapies. But it has also been known for a long time that the majority of cancers depend on the increased activity of a small number of proteins, the so-called oncoproteins. One of the most important oncoproteins is c-MYC. Under normal circumstances, c-MYC controls a variety of genetic pathways in the cell that are important for determining the rate of cell proliferation and cell growth. In 60 to 70% of human cancers, c-MYC is expressed at higher levels than normal, and becomes essential for sustaining the fast cell divisions on which cancers critically depend. If we could find a drug that interfered with c-MYC's activity, we would have a drug that would be highly effective against the majority of cancers. So, why do doctors not prescribe drugs against c-MYC to their cancer patients yet? Unlike so many other proteins in the human body, c-MYC is an intrinsically disordered protein. It does not take up a defined three-dimensional structure where scientists could identify suitable pockets to target drugs against. The structural flexibility of c-MYC is one of its most important functional features, which allows it to bind to many other proteins by adapting its shape in a complementary manner. Such proteins have been a challenge to drug. With advanced computer simulation methods, we can start to address the problem. For the first time, scientists from Imperial College London have been able to simulate the complete structure of c-MYC bound to DNA over extensive time periods. In simulations, running for several months on high-performance computers, they were able to watch the structure of c-MYC, including the intrinsically disordered portions in atomic detail. They made several unexpected discoveries, including regions of the protein that interact among themselves, and other regions that are surprisingly stable in their structure. These stable regions could indeed serve as target sites for small molecule drugs that prevent c-MYC from carrying out some of its functions in cancer cells. Considering that very little was known about c-MYC structure before, the new insights provide new ideas for experimental approaches that may ultimately benefit cancer patients. Dr. Jim Watson, who received a Nobel Prize for solving the structure of DNA, has this to say about c-MYC. A multiplicity of MYC-inhibiting specific drugs may have to be in our arsenal before we can routinely move beyond delaying death from uncurable cancers to true lifetime-long cures. Unfortunately, Google Trends shows clearly that the biomedical research community has lost a considerable degree of interest in c-MYC over the last decades due to the seemingly unsurmountable problems. The availability of new structural models obtained by high-performance computing reveals the most promising path to generate new ideas of inhibiting c-MYC. These ideas have the potential to become critical catalysts in this important cancer research area. c-MYC will pursue a two-pronged research strategy that tightly integrates experimental methods with computational biology. Initially, efforts will be mainly focused on structural biology projects to test the predictions from the computational simulations, and to initiate drug screening campaigns. Many of these efforts can be carried out through academic collaborations and contract research organizations. Because of their lower overhead costs, more and more biotech investors see such business models as ideal for maximizing the impact of their investments. c-MYC, the future of cancer therapeutics.